Hi everyone, and today we're going to talk about digestion. Digestion basically is the process of breaking up complex molecules. So far we've covered three complex molecules that our body uses. These are carbohydrates, C-A-R-B-O-H-Y-D-R-A-T-E-S, proteins, P-R-O-T-E-I-N-S, and lipids, L-I-P-I-D-S, or fats. On this side of the board, we have an example of these molecules. As you can see, this is a long chain of glucose molecules, and it is a polysaccharide. And we have here a polypeptide for protein, and we have fatty acids and glycerol for fats. These molecules are often too large for our cells to have them diffuse into it. So our body needs to use something called enzyme to break them down into smaller particles, or smaller molecules rather, so that they can be diffused. Let's look at this a little bit closer. Here we have a protein molecule. This protein molecule is very complex, and hence our body cannot digest this. Our body cannot absorb this as its large state. So our body will tend to break this down, and we will have two smaller molecules that can be then used to release energy. In most cases, whenever our body tends to break these molecules, water is released, and the process of breaking molecules and releasing water is called hydrolysis. Here is an example of hydrolysis in peptide bonds, because we're breaking a peptide bond in this protein. I'm going to review the three macromolecules that we talked about in the previous lesson. Again, it's proteins, and it's a very starch molecule, it's very large, and fat molecule. As I mentioned before, the building block of proteins are amino acids, and I'm going to use these shapes to represent a chain of amino acid forming a polypeptide. I'm going to use these shapes to represent carbohydrates, and I'm going to use this in the future to represent a fatty acid. These molecules, as you can see, they are too large for our body to diffuse, so we use enzyme E-N-Z-Y-M-E-S, enzymes, to break them down into smaller particles. After we use the enzyme protease, we can break down a protein to give all of these single units of amino acids. By using the enzyme carbohydrates, we can break down the starch molecule to give individual units of sugar or glucose. And fatty acids, we can use the enzyme lipase to break that down so that we can have the fatty acids broken away from the glycerol molecule. Again, this whole process of digestion is just to break the complex molecules into its individual forms. Proteins can be broken down to amino acids. Starch molecules can be broken down to sugar, glucose, for example, and fats or lipids, can be broken down to fatty acids. And in our lessons on enzymes, we said that enzymes basically speed up chemical reactions, and enzymes help to break molecules into smaller pieces. So if we have an enzyme, and here's our enzyme, and this is our macromolecule, the shape fits exactly, hence it will work. They then form an enzyme-substrate complex, and the enzyme breaks the molecule into two specific parts. If, for example, we have the same enzyme, I'm going to try to mimic this shape right now, and this is our macromolecule. Would the enzyme fit into the shape? No way. So it is not going to break this molecule into smaller pieces. It will not work. So that is why when we talk about enzyme, we say that enzymes are specific. They can only work on the macromolecule if the shape of the substrate is can fit into the enzyme active site. So where does all of these molecules, where are they broken down or digested? They're digested through our alimentary canal. Our alimentary canal starts with the esophagus. It continues down as food travels to our stomach. It then moves to our small intestine, and finally it moves to our large intestine. This should be a review from middle school. However, digestion, not all of the molecules are digested at the same place. Digestion actually starts in the mouth. A few molecules are broken down in the mouth. Then, in the stomach, digestion continues with another few molecules being broken down. In the small intestine, a few molecules being broken down. And here, the small intestine, majority of the molecules are broken down. And the large intestine is where the waste products that we do not need, is going to be moved to this section, and water is going to be absorbed, and then all of the waste substance passes out of the anus. This is a view of your alimentary canal from the front. And this is a view if you were to dissect a living organism, most likely a human being. You will see all of the structures are not so clearly separated, but they're all so put together and held together by membranes. Now that we have covered the basic parts of the alimentary canal and also what molecules need to be broken down, we're going to look at what specific enzyme work at which part of the alimentary canal. We start off with the mouth, and the enzyme salivary amylase would work to break down, you should notice this starch and disaccharide, and this you should notice that carbohydrates start to be broken down. When food moves into the stomach, pepsin, which is an enzyme, starts to break down proteins into large peptides. And in the small intestine, we have two parts of digestion. We have some of the enzyme that is released from the pancreas, and they are amylase, trypsin, and lipase. Amylase tends to break down starch, trypsin tends to break down proteins, and lipase tend to break down fats. The digestive process continues with maltase, sucrase, lactase, and peptidase. These few enzymes at the beginning continues to break down the disaccharides into monosaccharide, and peptidase continues to break down dipeptides into amino acids. Now, there's no way that I want you to remember the name of these enzymes. However, I want you to locate the part of the alimentary canal and what macromolecule is being broken down. So now that your macromolecules are completely broken down, what happens to this? Well, this is going to be representing your small intestine. Your small intestine has small finger-like projections that allows for molecules to be diffused through and enters your bloodstream. So now these small molecules are small enough to pass into your bloodstream, and then once it's next to the cell, it passes or diffuses into your cells. Before we move into the last section of our lesson, I really want to leave you with something to think about. Say, for example, we have a beaker, and in this beaker we have a semi-permeable membrane, and we add glucose and starch into this membrane. Which of these molecules do you think will be able to diffuse into the water? Do you think that glucose will diffuse, or would starch diffuse?